Hello guys, in this video I'm going to be talking about mitosis. Now humans grow, we all grow. We grow from a little zygote, which is just a fusion of two gametes, into a very big adult. So how does this happen? This happens by a process called mitosis. The growth happens by mitosis, whereas the division in the sexual cells, that is the sperm and ovum, happens by meiosis. Now let me first explain what mitosis is. Mitosis basically is the cell division in the body or somatic cells. Somatic cells contain 2n number of chromosomes, which means they are diploid. Diploid basically means there is a pair of chromosomes which you acquire from your both parents. So that is mitosis basically. Mitosis is also called as equation division because at the end of mitosis we get two daughter cells which both have 2n number of chromosomes. So it's just a division, but chromosome number is equal. So it's called an equation division. It's the interface which takes up the most time. In fact, 95% of the time required for cell division is actually taken by the interface. So the mitosis can be explained through this simple diagram. Now here the phases G1, S, G2, M, G0 are shown. Here the phases G1, S and G2 are collectively called as the interphase. The interphase is the phase before two successive mitotic phases. This is the phase where the cell prepares itself for the process of cell division. You know, the cell has to prepare itself. It has to grow larger in size. It has to replicate its DNA, you know, to give both the daughter cells the same amount of DNA. It has to replicate the centrioles and so on and so forth. And there's this another phase called G0 phase or quiescent stage or something like that. It's the stage where the cell doesn't undergo division. You know, there are certain cells like the neurons in the central nervous system and certain other cells which normally do not undergo cell division. So these cells are basically in the G0 stage. So the process itself can be divided into four phases, namely prophase, metaphase, anaphase and telophase. And these phases are collectively called karyokinesis. Now karyo means nucleus and kinesis means division. So in these four phases the nucleus is divided into two, and in the final process, which is the cytokinesis, the cell itself is divided into two, ending the process of mitosis. Now let me explain how the interface works. The interface basically is composed of three subphases, G1, G2 and the S phase. The S phase occurs in between the G1 and the G2 phases. This is how basically the nucleus looks during the interphase. Yeah, so during the G1 phase you cannot see any difference because the chromosomes are in their chromatin form and they are so tiny that you cannot see them. They are not condensed. So the cell is normally carrying its metabolic activities. Now the one distinguishing thing which can be seen is the growth of the cell. The cell has to grow to accommodate both the daughter cells. So the cell growth is seen, but the DNA replication doesn't occur during the G1 phase. Now the G1 phase is followed by the S phase where the synthesis takes place. S here is for the synthesis. Now DNA synthesis takes place, and now we have from 2n to 4n number of chromosomes. And there's also a duplication of centrioles. Now centrioles are very important structure when it comes to cell division. They basically form what we call these spindle fibers, which are very useful in pulling these chromosomes towards the pole region during the anaphase and the later stages of metaphase. So this is basically the S phase, and the G2 phase is characterized by the synthesis of new proteins and other cellular stuff which is essential for the cell division. So the interphase is now followed by the prophase, which marks the start of the mitosis. Now actually the mitosis occupies a small time for the total time required for the cell division, and the 95% of the time is actually occupied by the interphase. So that's why when you see a live cell under the microscope, you normally don't see it dividing because most of the time it's in the interphase preparing for the next division. So yeah, this is the prophase. the prophase is generally characterized by the condensation of chromatin into chromosomes. Now chromatin is a long thread connected by the centromere. Now this chromatin changes into a condensed form by shortening and thickening. Now this is called your chromosome, and the centrioles move towards the poles, because from the centrioles have to move towards the poles, because from that place they have to give the spindle fibers which are connected to the centromeres. Now let me quickly explain what centromere is. Now a chromosome consists of two sister chromatids like that. They're joined at the point called as centromere. Now the centromere contains a system of fibers known as kinetochore, which are very essential in the process of metaphase and earlier stages of anaphase. So yeah, that quickly let me explain what prophase does. It condensation of chromatin takes place, movement of centrioles take place, and it is seen that towards the end of the prophase, the nuclear membrane, which is the membrane which surrounds the nucleus, starts disintegrating. Not only that, but also Golgi bodies, endoplasmic reticulum, and the other small structures disappear, so that the chromosomes spread into the cytoplasm to start the next phase, which is the metaphase. Now the metaphase is the most important phase in mitosis, because this is the stage where the chromosomes can actually be seen as distinct chromosomes. Now whereas in the other phases it cannot be seen, as it exists in the form of rough chromatin. So this is a really important stage in mitosis. Now the metaphase is simply characterized by disappearance of nuclear membrane, as we saw in the later stages of prophase, and it is also characterized by the movement of chromosomes towards the equatorial plate, or what we call the metaphase plate. So it facilitates the movement of the sister chromatid towards the poles, connected by these spindle fibers. Now what spindle fibers are? They are these small fibers which come out of the centrioles, and they are connected to the kinetochore of the chromosomes. Now the chromosomes line up at this metaphase plate, and these spindle fibers are connected to the kinetochore. This is basically what happens during the metaphase. Okay, we discussed till the metaphase, where the chromosomes line up at the metaphase plate. Now comes the anaphase. The condensation of what happens in anaphase is the chromosomes that we talked about earlier, which line up at the metaphase plate or the equatorial plate of the cell, now are separated by the pull offered by the spindle fibers, and they move towards the opposite poles. So this chromosome right here, it moves towards this pole, and this one moves towards this pole. So yeah, okay, this one moves towards this pole, and this one moves towards this pole. So I've drawn it with colors here to understand where each one goes. So the black one here grows towards the upper pole, and the red one goes towards the lower pole. So the chromosomes actually move towards the opposite poles, and separation of chromosomes occur, and the movement of chromosomes towards the opposite pole occurs during the anaphase. Now the anaphase is followed by the final phase, which is the telophase. Now the chromosomes are now in the terminal region of the cell, so the chromosomes decondense, which is the exact opposite of what happened in prophase. In the prophase we saw that the chromatin condensed to become the chromosome, but now what happens is the chromosomes decondense to become the chromatin fibers. Now start losing your view of the chromosomes, and they become chromatin again. Now the telophase is also characterized by the formation of nuclear membrane, endoplasmic reticulum, and the Golgi bodies. So this is essentially what happens in the mitotic phase. So basically I'm condensing the entire mitotic phase. What happens is prophase is condensation of chromatin into chromosomes, the movement of the centrioles into the opposite poles, disappearance of nuclear membrane, nucleolus, and the endoplasmic reticulum and the Golgi bodies. Then follows the metaphase. What happens in metaphase is the chromosomes line up at the equatorial plane, and the spindle fibers, which are given out by the centrioles, connect to the kinetochore, which is present inside the centromere of the chromosomes. Now this is followed by the anaphase. Now the chromosomes move towards the opposite pole, and they are grouped at the terminal regions of the cell. And the final phase, which is the telophase, is marked by the decondensation of chromosomes and the formation or the reappearance of the nuclear membrane, endoplasmic reticulum, and the Golgi bodies. Now this is finally formed by cytokinesis, which means cell division. So there are basically, the cytokinesis happens depending on the type of cell. In plant cell, where the cell wall is inextensible, you cannot form a cell wall here. It takes place via a cell plate. Whereas in an animal cell, it takes place via a furrow. So basically a furrow is formed here and the cells divide. So at the end of mitosis, each daughter cell contains the mitochondria, all the cellular organelles, and they are equally divided. So this is mitosis, and it's only fair to call it equation division because the cells here have everything equal. The cells are identical to the parent cell, and that's the speciality of mitosis. And it's also in a sense kind of a disadvantage because you get the same, you get the daughter cell which have the same genetical constitution, so you cannot have variation in your organism by mitosis. So that's my mitosis video. Hope it helped. Peace.